Pinus. It comes under division Gymnosperms, class Coniferopsida, order Coniferales, family Pinaceae, and it includes about 105 species. 5 Indian species growing wild in Himalayan regions are Pinus gerardiana, which is also known as a Chilgosa pine, Pinus roxburghii, known as chir pine, Pinus wallichiana, known as blue pine, Pinus insularis, also known as Kashi pine, and Pinus armandi. External morphology. Pinus is a large perennial evergreen plant. The branches grow spirally and therefore it gives an appearance of conical or pyramidal structure. The sporophytic plant body is differentiated into root, stem, and leaves, which are acicular, that is needle-like. The taproot with few hairs is present, but it disappears soon. Later on, many lateral roots develop, which help in absorption and fixation. The ultimate branches of these roots are covered by a covering of fungal hyphae called ectotrophic mycorrhiza. The stem is cylindrical, erect, and remains covered with bark. Branching is monopodial. 2 types of branches are present, namely long shoot and dwarf shoots. These are also known as branches of unlimited growth and branches of limited growth. The long shoots contain apical buds and grow indefinitely, and many scale leaves are present on the long shoots. The dwarf shoots are devoid of any apical bud and therefore they are limited in their growth, and these arise on long shoots in the axil of scaly leaves. A dwarf shoot has 2 scaly leaves called prophylls, followed by 5 to 13 cataphyls arranged in 2 by 5 phyllotaxy and 1 to 5 needles. But the number of needles varies in different species. In Pinus monophylla, there is only 1 needle and therefore it is unifoliar. In Pinus sylvestris, there are 2 needles and hence it is bifoliar. In Pinus gerardiana, there are 3 needles and hence it is trifoliar. In Pinus quadrifolia, there are 4 needles and hence it is quadrifoliar. In Pinus wallichiana, there are 5 needles and hence it is pentafoliar. This is Pinus monophylla, Pinus sylvestris, Pinus gerardiana. And now coming to the branches of limited growth or the dwarf shoots. These are present on the main stem and on long shoots in the axil of scale leaves. The dwarf shoots possess many scale leaves and bear a group of foliage leaves of needles at the apex. It has a terminal ephemeral bud and covered with scale leaf, that is cataphyll, in Pinus wallichiana. Coming to the branches of unlimited growth or long branches. Here the growth is indefinite by apical bud and it arises as a bud in the axil of scale leaves. The older parts are covered with scars of left by the fallen dwarf shoot subtending scales. Younger parts bear brown scale leaves with dwarf shoots in their axil. Coming to the reproductive structure. Pinus plant body is sporophytic and Pinus is monoecious, and hence the male and the female flowers are present in form of cones or strobilus on separate branches of same plant. Many male cones are present together in form of clusters. These develop much earlier than the female cones. Coming to the male cone. The male cone develops in cluster in the axil of scaly leaves on long shoots, and they replace the dwarf shoots of long shoot. Each male cone is ovoid in shape and ranges from 1.5 to 2.5 cm in length. A male cone consists of a large number of microsporophylls arranged spirally on the cone axis. and each microsporophyll is small, membranous, brownish colored structure. A microsporophyll consists of a stalk with a terminal leafy expansion, the tip of which is projected upwards and is known as apophysis. Two pouch-like microsporangia, that is pollen sacs, are present on the abaxial or undersurface of each microsporophyll. In each microsporangium are present many microspores which are winged. Next, female cones. The female cones of Pinus are also called as ovulate stabili. They appear in the month of February in plains and in May in hilly regions. Each female cone arises in the place of dwarf shoot, and each cone takes 3 years to get mature and set the seeds. The first year cones are comparatively small, compact, reddish green in color, whereas the second year cones are comparatively larger and they become slightly woody. The third year cones are large and woody and develop seeds at the end of the third year. They are elongated, cylindrical shape and 15 to 20 centimeter long. Coming to the anatomy of root. First, anatomy of young root. In a transverse section of young root, the outermost layer is the epiblema, which is having thick walled cells with many root hairs. Epiblema is followed by many layers of parenchymatous cortex. Inner to the cortex is present a layer of endodermis and many layers of pericycle. Vascular bundles are radially arranged and di-to-tritarch with exarch protoxylem. The protoxylem is bifurcated, that is Y-shaped, towards the periphery, and in between each bifurcation is present a raceme canal. Phloem is present alternate to xylem, and pith is poorly developed or totally absent. Coming to the old root. In the old root, the outer layer is cork, which is formed by meristematic activity of cork cambium. This cork cambium cuts off secondary cortex towards inside. Many raceme canals and stone cells are present in the secondary cortex, the cells of which are separated with intercellular spaces. Below the phloem develops cambium, which cuts off secondary phloem towards outside and secondary xylem towards inside. Crushed primary phloem is present outside the secondary phloem. Many uniseriate medullary rays are present in the secondary xylem. The primary xylem is similar to the young root, that is, each group is bifurcated, that is Y-shaped, and a raceme canal is present in between the bifurcation. Coming to the anatomy of shoot. First is the transverse section of long shoot. Many leaf bases are present on stem, due to which it appears wavy in outline. The outermost layer is single layered, made up of thick walled cells, and this is the epidermis, and a cuticle is present above the epidermal layer, and this is followed by multi-layered cortex. A few outer layers of cortex are sclerenchymatous and some inner layers are parenchymatous. In the inner layers of cortex are present many raceme canals. The stele is ectoploic siphonosteal. Vascular bundles are conjoint, collateral, open and endarch, with 5 to 10 vascular bundles arranged in a ring. Endodermis and pericycle are indistinguishable. Narrow xylem rays connect the cortex and pith. Endarch xylem consists of only tracheids. Phloem is present on the ventral side and consists of sieve cells, sieve plates, phloem parenchyma and some albuminous cells. Intrafascicular cambium is present between xylem and phloem. Many leaf traces are also present, and a small parenchymatous pith is present in the center of the stem. Thank you.